So it's my pleasure and honor to moderate this panel of immunotherapy at Sinai Innovation today. And on the panel, we have three exceptional immunotherapy experts who have significantly contributed to shaping current immunotherapy strategies against cancer, but also infectious diseases. So we are very excited to have Judy Lieberman with us today. Judy is a professor of pediatrics and the chair of the Executive Committee of Immunology at Harvard Medical School. Dr. Lieberman has received her PhD in physics from Rockefeller University and then worked as a theoretical physicist at the Institute for Advanced Study at Princeton prior to receiving her medical degree from Harvard and MIT. What a pedigree. She designed and led early T-cell immunotherapy trial against HIV, and today her lab continues to study the molecular pathway used by cytotoxic T cells to kill their target cells, especially those activated by enzymes and perforins. Judy has also been at the forefront of the RNA silencing field, promoting its use as a research tool to learn about basic biology, but also learn about pathophysiology, and was one of the first to show in animals the therapeutic potential of RNAi to treat infection and cancer diseases. With us today, we are also fortunate to have Carl June. So Carl was introduced by Steven Burakov. So I'm going to be very brief. Carl is the director of the Center for Cellular Immunotherapy at the Abramson Cancer Center at UPenn, and he's an investigator of the Abramson Cancer Research Institute. Together with Michel Sadona at MSKCC, he was the first to use gene therapy to enforce the expression of CARs in T cells and use CARs T cells to treat patients with cancer. In 2012, CAR T cells developed by Carl June's lab successfully treated a young girl with severe relapsed acute lymphocytic leukemia, with more success reported as the trial progressed. That same year, the pharmaceutical company Novartis licensed the rights to therapy created in June's lab, and in the summer of 2017, Novartis received the first ever FDA-approved for CAR T-cell therapy called Kymriah, that Carl, in fact, reviewed today, for the treatment of children and young adults with relapsed and refractory B-cell ALL. And joining us from Sinai, we have my wonderful friend and colleague, Nina Bhardwaj. Nina is professor of medicine, and she's also the director of the cancer immunotherapy program here at Sinai. Nina obtained her MD PhD from NYU and did her internal residency at the Brigham. She trained in rheumatology at the Hospital of Special Surgery. She then worked at the Rockefeller with Nobel Prize Ralph Steinman, examining how dendritic cells can modulate anti-cancer and antimicrobial immunity. Nina was one of the first to develop DC-based vaccines for the treatment of cancer and HIV patients, and more recently, she has been actively involved in the development of neoantigen vaccines for the treatment of cancer patients. So thank you all for joining us today. It's a privilege to have you and to have your thought and perspective on the prospect of immunotherapy for cancer patients. I'm going to open the panel for discussion. I will be asking the first question to get this moving, and then I'll be happy to take questions from the audience. And my first question is for Judy Lieberman. So Judy, your laboratory has made seminal contribution in the field of RNA interference, and we certainly remember the big excitement 10 or 15 years ago around RNAi therapy, with Merck acquiring, I think, Sinai Therapeutics for over $1 billion. So that's reminiscing of what's happening with CAR T-cell therapy today. So since then, there has been many clinical trials with some clear successes. And we'd like, in fact, to hear your thought of the prospect of RNAi therapy. What are the big hurdles? Can we overcome them? There has been some recent, very exciting results from this phase 3 trial in using RNAi for the treatment of amyloidosis. So can you summarize for us what's the future of RNAi? And maybe if you can comment of whether RNAi will survive CRISPR technology or coexist with CRISPR. You know, do we still have a space?
In 2001, and this past month, the results of a phase 1 trial for gene knockdown in the liver were announced, and the results are, it's taken 15 years to get to where we are now, but this is a new type of drug, it's a new platform. I think we're, And the clinical trial results are truly astounding. The p-values for significance for the major endpoint were 10 to the minus 24. All the secondary endpoints had similarly amazing results, and it's proven to be relatively safe. So that's to knock down genes in the liver. If you want to treat cancer or use RNAi for immune modulation, it's a bit of a challenge. You have to figure out ways of transfecting in vivo cells that are harder to get to. T lymphocytes, for example, are probably the hardest cells to transfect in the body. We've actually worked out ways of doing that, both for immune cells and for cancer cells. So I think it's going to be possible. It's just going to take a little bit more time. I think in general, we're entering a new era of nucleic acid-based therapies, that gene therapies like CAR T cells. This week, there's a new gene therapy for blindness in the eye with Catherine Hai's company, which was approved. We have the first antisense oligonucleotide therapies that have been blocking splicing. So basically, everything that we've been learning in the molecular biology revolution of the past 35 years since Carl and I started working in this, is finally, after a lot of effort, beginning to bear fruition. And I think that there are going to be new classes of drugs. Antibodies will be, will maybe replaced by nucleic acids, or certainly we're going to have a lot more tools in our armamentarium.
Thank you. So can you comment about CRISPR? Are they going to be competing?
Yeah, well, I think, there are things that, CRISPR is for permanent editing of the genome. So that is attractive if you have, say, a primary immunodeficiency disease and or some primary genetic defect for correcting it. I think as of now, it's a little bit scary because we don't have complete control over where we edit, that there are translocations and things that still occur, although a lot less than the first generation. But I think for genetic type diseases, that's going to be the way to go. One thing about RNAi, which you may not realize, is that the chemistry has been improved to such a degree that now gene knockdown after single subcutaneous injection can last for 6 months. Wow. For cancer cells that are rapidly dividing, it's not going to last that long because the RNAs will get diluted. But I think there'll be different indications. I think there'll still be a role for RNAi, but certainly CRISPR is going to be amazing in the future.
Yeah, Carl?
So what Judy said is very similar to my field, where the ideas really are not new. I mean, what's taken longer has been the details to make it actually work, so the translational issues. But I mean, when Judy started out, the RNAi, it worked in small scale, but to get it to work as it does now took a lot of seemingly small technological improvements. In the area of gene transfer with vectors like we did, it's the same thing. We started off with retroviral vectors that were more prone to silencing, and then many stepwise improvements made for better and prolonged expression, less silencing, and so on. So I think one lesson is, the ideas really were all there 50 years ago in many of these cases. And in the gene modifications, they were certainly there 20 years ago. It's taken until now until they're getting FDA approved. And it really is a watershed for the whole field to see that what they could do in a liver knockdown like that, and that data was stunning. And there were many failures before, but it wasn't that they didn't have the ideas.
Brian, to ask a question?
Thank you. Yeah, you I guess this is to Dr. June. So you had detailed before all the things that Novartis had done to sort of bring it to bring this to trial and to the world, around the world. But how much of it was driven also by money, public money? How much of the innovation, how much of the development was there, since you sort of watched it be taken forward? Because we're often heard, or being told that the price of these drugs is sort of driven by how much investment the pharma put in. But then I think I at least saw a lot of investment going from the public in the years that the drug companies wouldn't invest a single dollar into these things. So maybe you can, since you watched it firsthand, you can tell us a little better about that.
So, it's a complex issue. I'll tell you what Novartis has gotten FDA approved is what we made in my lab in 1992. So they did not improve on it at all. What they did was scale it out and do what we did in one patient or in small scale. They made it global. And pharmaceutical companies are good at that. That's something academics aren't very good at. And so innovation, in general tends to happen at universities, academic centers. And I think that's especially going to be true in cell and gene therapy, where, for instance, the way you find out if one of these therapies work really depends on patient biopsies and a careful analysis. So it used to be we had, for instance, when targeted therapies were made, like Novartis had Gleevec in 1999. What happened was a kinase inhibitor was made, imatinib, and then the trials were done, and then the pharma, BMS, and Novartis all made second-generation kinase inhibitors that got better and more specific and could hit the different mutations where there was resistance, and it never went back to academia, really. It was all that drug discovery and medicinal chemistry was well housed in pharmaceutical expertise. But in the case of, I think, cell and gene therapies, it's really at the academic centers. And so we're going to see a very different history. It's going to be more partnerships as things develop between academic centers and pharma and biotech. That's one difference. Another is, it's often been said, the U.S. pays for drugs around the world. We develop drugs here in the U.S. We have NIH funding, it's governmental funding, and then other countries benefit off that. Often the prices are lower at other countries, and so that's a geopolitical issue. And so we have this issue now that's brought up. For instance, most of the development of CAR T cells was actually all done off philanthropy and NIH funding, not the pharmaceutical investment. So then it becomes a political issue of how then should the U.S. healthcare beneficiary have to pay for that. And that's a very big discussion. I think that's one we all need to have.
Jimmy?
So I have a specific question for Dr. June and then a more general question for the panel. So we often say that tumors are the smartest immunologists because they figured out every way to evade the immune system. So the specific question for you, Dr. June, is going back to Julio's question. In any of the relapses, have there been relapses in which you've had, for example, a downregulation of the CD19, the way after allotransplant you can get relapses with downregulation of HLA molecules? And can you, has that happened? Can you predict it? And then the more general question for the panel is, what are the, in your opinions, what are the key elements of immune monitoring, both in terms of risks and then in terms of the efficacy of therapies that we need to begin to incorporate into our clinical trials?
Okay, so I'll start off by saying, there's, when you have, gene marked cells and with these immunologic manipulations, the ability to understand resistance is much easier than many small molecules. And one of the analogies is, for instance, taxane resistance. It's been recognized for 20 years now, develops, and yet there's been really no fix, no good way to overcome that. It's been a really tough nut to crack. within 2 years of treating CD19 positive ALL, what was recognized is the most common form of resistance is CD19 loss. It turns out all antibodies to CD19 bind to exon 2 of CD19. And CD19 has 14 exons, and it turns out that most, the tumor, as you said, it's smart, they have, the mechanism of relapse is exon skipping. So there's what's called a 1,3 splice variant expressed in these ALL cells. And so the cell still has, CD19, it can still bind to the BTK signaling apparatus, but it no longer has a target of the CAR. It's a really elegant escape, very similar to some of the things done the HIV virus does, for instance, if you give single antiretroviral therapy. So that's it's a splicing defect that wasn't really predicted. Now, if you go to RefSeq, it turns out in CD19, there is, in fact, a normal transcript can be found in B cells that has this 1,3 splice variant. And no one's ever found a function for that. There's now people thinking it may have a role in early B cell selection, where you could have CD19, but it's not fully active, and it becomes more intracellular expressed. So this is one area where we may actually learn new biology now about B cell development than had been thought before, because we see this. Now, the other aspect about this CD19 escape in ALL is this is pre-B cell ALL, is it's not seen in CLL. So CLL, as you know, is a more differentiated B cell that gets, through different driver mutations and so on occurs. But there, we haven't seen CD19 escape like this. So it's a phenomenal ALL. I think we'll see this in many different tumors, and that we're really lucky that in most cases B cells really are addicted to CD19, and they have a hard time losing it.
Yeah, I have a question. CD19 is a signaling molecule, and it has an advantage to the tumor to keep it expressed, which may be partly why you don't get so much escape. My question is, I'm not familiar with mesothelin, the antigen that you've chosen for your tumors. Is that similarly a kind of signaling molecule, and would you expect to see more escape in that case?
So, the answer is we don't know. We think mesothelin is, there is some gain of function in tumors, because, it's a ligand for CA125, and tumors overexpress it for reasons that aren't totally clear. There's some data that helps in their signaling. So, but we still don't know really, is it safe to drug mesothelin, meaning will the normal cells, will there be toxicity that's acceptable, a window of, or not? I mean, we knew with CD19 that it would be acceptable because kids could be born without CD19, and it was a tolerable, as you know, immunodeficiency. So it's still open. The one safe target we know with solid tumors with CAR T cells is EGFRB3. That's a splice variant. There's a novel glycine that gets introduced and that loses exon 2 to 7, and it's different than EGFR wild type, and it's not expressed on normal cells. But beyond that, we don't really know. Now, Jamie also asked about monitoring, and this is a big issue that I think academic centers, it's a real opportunity for high-end academic centers, because finding out how patients are resistant or not to therapies, can allow you then to treat with a higher probability of success, but also discover new mechanisms of resistance. The drug companies, biotech and pharma, traditionally aren't really interested in this much, and they tend not to really fund it in experimental protocols. So if you have an immune monitoring system, and I happen to know you do here, that's quite good. That is a real plus, because now Novartis is going to be selling these cars, Kites are going to be selling them, and we have very careful monitoring set up in our center in Philadelphia, but on the commercial scale, that's not going to happen, and the clinicians are going to be flying by the seat of their pants. We really use the data on are the CARs still functioning? Is B-cell aplasia there or not? And as a way to instructively tell us what to do in different situations. So right now, that's a big gap, and I think it's going to get larger.
And I'll just add to that, I think there's a real need. I think in your case with the CAR T-cell therapies, you have the T-cells you can monitor and obviously the targets, which are readily accessible. It's in the solid tumors that the monitoring becomes much more complicated. And so advocating for solid tumor tissue access, for example, pre and post intervention is important to get a sense of the genomic landscape of the tumor. What are the mutations? What are these drivers or non-drivers? Do they elicit neoantigens, for example? And then uncovering mechanisms of resistance. And we're beginning to understand that mechanisms of resistance are huge. And you had a slide actually highlighting some of these, but going from just loss of class 1, beta 2 microglobulin mutations, interferon signaling, stromal elements, EMT transition, et cetera, not to mention cytokines and all the metabolism, metabolic dysregulation that occurs in the tumor. And this is going to vary from tumor to tumor. So getting a better picture of what's in a tumor, I think, and improving our methods of monitoring are going to be key.
I couldn't agree more. I think, I mentioned, we were lucky starting with leukemia, CLL and ALL, because every patient standard of care gets a bone marrow biopsy a month later. And we could then really get good biomarkers of correlates of efficacy and failure and so on. But now, we have two things fighting against. In solid tumors, generally, the bias at the University of Pennsylvania at our IRB has been on phase 1 trials not to allow serial biopsies. Unless you have melanoma, where it's basically a skin biopsy, and that you can do ad lib. But in solid tumors, basically pancreatic cancer patients get an initial biopsy, and then oftentimes the rest of their life, they never get another tumor biopsy. So I think we as oncologists and all need to change that standard of care, because it's almost, I think, unethical to conduct these trials without biopsies, because you learn why it worked or didn't work, and then that means we don't treat people futilely.
Yeah.
And then, so futile therapy will become less, and we'll more rapidly get the answers to why things work or don't work. So I think we need, it's, find a way that every trial gets biopsies on the patients.
Judy?
Yeah, and just to add to that, I think it's not just uncovering mechanisms of resistance, but probably finding new avenues for treatments. So even traditional foundation type medicine type analyses, further investigations are suggesting that patients with some of the mutations like BRCA1 or whatever can be associated with a higher mutational load. So perhaps combining targeted therapies with checkpoint inhibitors that may be very patient-specific is also going to be important and will come as a result of sequencing these tumors.
Yeah, I'd like to make a comment too. So I think we're in some ways of the successes of immunotherapy, and most of the tumors and modalities we have given dramatic successes, durable cures in subsets of patients, but the majority of patients we still don't have success with. And I think that the manipulating the immune response, especially for solid non-hematologic malignancies, is incredibly challenging. And our assays, even in mice, are not that sophisticated for figuring out what's going on in these very complex microenvironments. I think it's going to require new technologies. Last week I heard a seminar where Hitaplu has basically figured out ways of using very small camelid antibodies to do PET imaging, where he could actually distinguish in mouse tumors whether the T cells that infiltrated the tumor infiltrated diffusely throughout the tumor or not diffuse. So I think it'll be possible to develop new non-invasive technologies which will really give us new kinds of ways of looking at what we've done and how we can do it better.
Absolutely. Immune monitor, just to continue on this, I think also access to fresh tissue instead of only bank tissue is going to be very important. So developing new paradigms of accessing and working on live tissue is very important. Ben?
So I'm struck by the many parallels between the cancer therapy and the HIV therapy and the antigenic diversity and the issues brought up about escape. And I was wondering if the panel could comment a little bit about a path forward with regards to targeted therapies, which might be more vulnerable to escape, and the more general immune modulatory therapies. And will there be a need to have multiple targeted, like combination therapy for CARs, for instance?
Judy and I both, I mean, she's done work in breast cancer and HIV. I've done work in HIV, and so they phenocopy each other in many ways. So at one level, they both cause immunosuppression. You know, I mean, HIV kills CD4 cells, tumor inactivates them, usually exhaustion mechanism. So different ways to get to the same endpoint, which is a non-functioning CD4 cell, is one example. I mean, the high mutational rates, we knew about HIV and the infidelity and so on of the polymerase, and it's only recently become available, you know, as to how unbelievably complex the subclonal diversity of tumors is. So they're very much that way. So that brings up issues of, you know, likely for cancer therapy with rare instances, we're going to need combination therapy, just like HIV. Another issue is oncologists in general like to do sequential therapies. I think that, you know, what we need to do is learn from the HIV field where you give combination. We have issues with, you know, so-called novel when you put in things and getting the FDA to approve or our IRBs, you know, but, you know, combination therapies. But, you know, all our science says that's unless you want to select for resistant variants, you know, and rather than sequential therapies to try to do combination. But it's really a clinical challenge in many cases.
The good news is that we have so many combinations to choose from, and as Carl said, the challenge is going to be how to combine them, and this is where learning about individual tumors and how to apply these combinations together is going to be essential, and I totally agree, combinations are going to be the key.
William?
My question is a little bit along that line, which is checkpoint inhibitors, of course, came onto the scene. We haven't really talked a lot about them as in terms of the transformative nature of how they've really taken diseases like lung cancer, which were really tremendously resistant to chemotherapy and standard treatments. And we see these very solid 30% responses, 40% at best. And it looks like we're starting to hit a wall, at least with the currently available checkpoint inhibitors. So my question to the panel is, how are we going to break that? Obviously, we heard a lot of exciting things about, in the heme malignancies in particular, about CAR T cells. But how do we get past what appears to be a wall with the checkpoint inhibitors that we have now?
Judy?
Yeah, so in my opinion, the main problem is that most tumors aren't well recognized by the immune system. Either they were never recognized or they've been edited out, the antigens. So I think, I think CAR T cells bypass that problem because they're supplying an external antigen, but then we discussed there might be resistance. So I think the question is how you can combine, what you could combine to get the tumors recognized. For me, that's really the central problem. So, you know, I think vaccination is an attractive strategy. Recently, there were some papers published in Nature with peptide tumor vaccines or mRNA tumor vaccines which were also looked like they showed the same kind of dramatic effects. But what was interesting was that, at least in one of those papers, when the vaccine didn't work and they added a checkpoint inhibitor, it did. And I think Carl's data today about adding Pembro to CAR T cells, you know, the immune response is so complicated that you have to really dissect it, see what way combinations will make sense. And, you know, there is a real danger for combinations in that the checkpoint receptors are part of the natural T-cell activation response, and they're there for a reason to keep things under control and not get out of hand. And I think when we've seen some combinations, they you do get into serious problems. So it's going to be like a matter of real intelligence to figure out the combinations, how you give them, whether you give them up front or sequentially, what order.
Yeah.
But it's an exciting new world where we have all these tools.
So Nina,
Just
maybe also
to-
you can comment on DC vaccine a little bit. So I think it will be important to put them in perspective. They have done some successes, not so much. Are they going to be revived now with this combination opportunities?
So I guess I could talk a little bit about cancer vaccines in general.
Yeah. Right.
Or the lack of cancer vaccines, I should say. You know, we only have very few approved therapies in that area in cancer. So we have BCG for bladder cancer. We have imicamod, which is TLR7 agonist that's applied to topical skin tumors, and Sipuleucel T, which is partially enriched dendritic cell cancer for prostate cancer, vaccine for prostate cancer that really didn't meet the mark that we'd hoped it would. It was given in castrate-resistant prostate cancer. It's still the only really approved cancer vaccine here. And I think in general that's in part because cancer vaccines have gone into very late-stage disease, where all these issues that we've talked about are not really understanding how to generate an immune response effectively on the part of the T cell, really disagreement over general adjuvant platforms, how to induce good immunity, and then the issue of large tumor burden and complicating and turning off the immune response. So that said, I do think we still have a need to understand vaccination better and how to do it better, and papers you were referring to with the neoantigen vaccines, very encouraging, and those were in the adjuvant setting, where we're beginning to understand that tumors with high mutational burden can, in some cases, translate into the presentation of totally new proteins that can result in a very efficient immunogenic response. MSI tumors responding to checkpoint inhibitors are a very good example for that. So, so I think the time is ripe in the right setting to sort of reinvestigate vaccine platforms, the types of antigens, neoantigens, possibly shared neoantigens as well, which we know also exist, in combination with checkpoints and other therapies. As far as dendritic cells go...
Combination with CARs also? Or no?
Possibly, yeah.
To
there are
re-expand
MUC1
and reactivate.
vaccines that are in the clinic. That's another possibility. And certainly mesothelin vaccines as well. And as far as dendritic cell vaccines, you know, we're still working on those. We're beginning to get a better handle of what subsets of dendritic cells exist now. In humans, we used to think of them as just 3. We now know they're probably at least 6.
Yeah.
And understanding the relative contribution of each of those dendritic cell subsets to vaccination is going to be key. One of the neoantigen vaccines that was reported on earlier was a dendritic cell-based vaccine given intravenously. So we also have to understand how to give it. That's just from Penn. I think there's still a lot more work to do, and clearly we haven't given up using that as a cellular therapy. And with gene therapy now here, to make this a better cell in the future as well.
Yeah, Julio?
I have two kind of general theoretical questions. There was a recent case report of a patient that had one type of childhood leukemia, And after cure, 22 years later, he developed another leukemia. They both were sequenced, and they shared truncal mutations. And it was suggested that a pre-malignant dormant clone persisted in the patient. So this, and the notion that there's a lot of the mutations that drive the disease are already in the pre-malignant disease. Has this made you think about using immunotherapy in the earlier stages, since there's a growing population that is detected as we age with these pre-malignant clones? So that's question one. Question two is, if you look at the tumor in response to immunotherapy, have you seen any epigenetic reprogramming of the tumor cells, either in leukemia or in solid tumors, in response to the cytokine storms that you generate that could cause evasion without really a selection, but rather through an adaptation? So those are my two general questions.
So, I mean, it's a pretty sobering thing to hear about a 22-year late relapse and shared truncal mutations. I think, so a subset of patients, do have, some, they have DNA repair mutations, and I don't know if that was looked at this patient, whether there were, PAL-B or BRCA mutations or something that, or whether, but I did mention, I think the reason bone marrow transplant, it's rare in AML to see a relapse after 2 years. And that, most likely when it's an allo transplant, not with autologous, and that's likely to ongoing immune effects that, keep these to be rare. Clearly moving immunotherapy up front, even to the point where, we have clinics, I'm sure you do here, of women with DNA repair defects, familial history of breast and ovarian cancer. Can we get to a point where we vaccinate them rather than have surgical ablation? And other people who have DNA repair, it's 20% of men with metastatic castrate-resistant prostate cancer have BRCA1 or BRCA2 mutations. It's incredible. No one had any idea of that a year ago. So can we, identify these people and then treat with immunotherapy up front with either vaccines, would be the most cost-effective. And I think upfront therapy with CAR T cells is going to happen more rapidly than we thought, because it took 50 years for oncology to develop what is a successful therapy for ALL in children, but it's extremely, there's a lot of morbidity, both in IQ, linear inches of height loss because of radiation and 2 or 3 years of chemotherapy. And I thought because of the conservative pace of physicians that, that to roll back therapy would take a long time. It turns out, the patients are pushing this at a way now because of internet things much more rapidly. So they are already in the Children's Oncology group designing upfront therapies with CAR T cells. And because now of our great genetics, you can predict that people who have, poor outcomes, with standard therapies, so they can be upfront treated with CAR T cells and or abbreviated chemotherapy, and then see if that has less long-term toxicity and just less overall morbidity. So I think immunotherapy in general will move up front. There's beautiful data from Hopkins now and BMS in non-small cell lung cancer using neoadjuvant, just between a biopsy before surgical resection of patients with tumor, just 2 or 3 weeks of PD-1 and or IPI, and they have amazing pathologic responses, after 3 weeks. So clearly that's where we have to go, and we oftentimes treat too late. So that's our challenge now, is to move things up front.
And if I could just add, so for example, in melanoma, anti-CTLA-4 is approved in post-resection stage 3 melanoma, clearly with an improved overall survival. And recent studies have suggested anti-PD-1 in the same setting will be even better and will probably be approved next year. And we're seeing now in women with cervical intraepithelial neoplasia getting DNA vaccines encoding HPV, a better clearance rate. So all these things are happening, and we are going to see things getting moved up, as Carl said.
And,
So
you know,
maybe
as far as your issue or question on the epigenetics, we really don't know anything yet. I mean, epigenetic modifiers given, like EZH2 pathway, for instance, stuff, may have a good way to change the balance between effector cells, Tregs, and so on. And we don't know what cytokine release syndrome does to the host. We think, so we have data that's not fully published yet, but in cytokine release syndrome and ALL, the cells making the most IL-6 are not the CAR T cells, it's the macrophages. And we think the innate immune system is probably doing a lot of the anti-tumor effects with CAR T cells. And so we need to broaden out beyond just looking at the T cells.
But just maybe a segue on this combination therapy. So as important as combination is probably going to be moving to the neoadjuvant setting to really target the low tumor burden. And this is also an important, I think, a statement to make, is that all these immunotherapy are really being tried a bit too late. We will gain in terms of tumor response if we were to move them as early as we can.
Yeah.
Charles.
So is there anything about the exceptional responders or the non-responders about the host? In other words, do people with better immune systems tend to respond better, or people with worse immune systems tend to respond worse? So thank you.
I'll say one thing. I mean, so we found, you know, this one in the one case I mentioned with TET2. So a function of the cells being central memory cells is what correlates with long-term responses. So we know with aging that, so pediatrics, their immune system in general has better function than elderly adults. And whether you look at healthy elderly adults and vaccinate or not, you get, you know, less robust results than if you look at later stage patients. And I think Jimmy Carter is an amazing, if you look at, you know, exceptional responders, how did someone who's over 90 years old have a complete response to metastatic melanoma? So clearly he's an outlier. And studying people with immune systems like that is really important. And I think what we'll find is that, I mean, some people have less senescence in the immune system, and so regenerative medicine becomes a big issue then.
Yeah, I actually just heard Mark Davis speak recently, and he's been just genomically profiling healthy individuals and then following them up for several years for whether or not they develop an inflammatory disease, diabetes, or cancer. And very interestingly, finding that I guess what he's calling, and you can correct me if I'm wrong, sort of genomes that confer, or this is from blood, signatures rather, that confer a high baseline inflammation status are correlated with a higher incidence of getting ill later. That's a very simplified version of what he said, but I think these sorts of studies are going to be very helpful for us to understand what the immune system, the imprint we have of our system, is going to do in the future.
Okay, the last question by Ramon.
Yeah. So I was just at a talk by Tony Rebus, and he was showing some data in melanoma using our herpes oncolytic virus in combination with checkpoint therapy. And in his talk, he kind of was provocative, and he said, you know, pretty much any type of vaccine that could, like a toll receptor agonist, neoantigen, virus, could potentially be equivalent. And I'm just curious how you think, Nina, for instance, do you think that that's a reasonable comment?
I think it's one way to get the immune system going, definitely, and you're probably referring to as intradermal TVAC plus anti-PD-1 combination, which looks very good. And so the idea is to really inject immune modulators into the tumor, which is what this virus essentially is doing, plus cytokines, to sort of in situ prime, mobilize the immune system, locally draining lymph nodes, and that together with checkpoint inhibitors will further expand it. And the presumption is you're vaccinating to tumor-associated antigens and neoantigens there. That's something we do here a lot. I don't know if Josh Brody's in the audience, but Josh and I do a lot of intradermal immune modulator studies here with the same concept and idea to do that. And, it certainly has worked, so.
And a follow-up question is, if the tumor that you're injecting into has a microenvironment that's so toxic for, an inflammatory response, how do you choose the tumor to do the vaccine in?
Yeah, it's a good question, because certainly we've had patients fail that therapy. And in the little bit that we've done, at least here, in patients who haven't responded, we're seeing a failure to respond to the intervention. No interferon signaling appears, no T cells come into the tumor microenvironment. Instead, you make these chemokines that draw in these immunosuppressive macrophages. So understand, this is why it also does become more individual. What tumors fail at this kind of intervention, and so a better understanding that will be necessary to see what else we can do to change that tumor microenvironment.
All right, so I think we are going to close the panel. I'd like to thank again Judy Lieberman, Nina Bhardwaj, and Carl June for sharing their expertise with us today. Thank you.